Good evening, everybody. My name is Sriram. I go by Narsi for the ease and convenience of all parties involved. I'm a design and development engineer at Hesperos, an organ-on-a-chip company located down in Orlando, Florida. And over there, we mostly focus on looking at multi-organ systems, looking at functional measurements from the multi-organ systems, and looking at forming PK-PD relationships. The talk that I'm going to be looking at today is biodistribution and PK modeling in a three-organ system, which consists of a GI tract, blood-brain barrier, and the central nervous system. Primarily, we're looking, in this case, is the PK model to look at transport across the GI and the BBB. Before I jump into it, I'd like to give credit to the tissue engineering team back at my company, so Dr. Mridhu Malik and Sarah Lindquist, who collected a lot of the data and developed the cell culture models and did a lot of the experiments. So moving on. Over at Hesperos, what we like to focus on is looking at multi-organ systems interlinked by a single fluidic path where they share a common recirculating medium. The medium itself is serum-free and usually, so the same medium for all the different organs. Looking at the surface chemistry itself, we try to focus a lot on the surface chemistry to establish extracellular matrix that's relevant, and also patterning the surfaces that we use to create a structured cell construct, as Dr. Schuler was talking about this morning, instead of 2D layers that are not structured. So if you look at the top right of it, oh no, that was the wrong one. There we go. If you look at the top right over here, it's a representation of the most common system that we work with currently, which is a 4-organ module. The organs that are included right now are the liver. We have cardiac cells, skeletal muscle, again cardiac cells, and neuronal cells. You might ask yourself why we have cardiac cells 2 times. So like I mentioned earlier, we focus a lot on the functional measurements. What are functional measurements? A lot of times when we're dealing with microphysiological systems, we're looking at endpoint assays, or essentially your readout, which would be your pharmacodynamics, which is what is the drug doing to your body, or in this case, what is the drug doing to the organ constructs in your system. We try to develop functional outcomes that you can correlate to a clinical outcome. So we use a lot of engineering principles here. We use microelectrode arrays to look at the ECG equivalent in vitro, and then an EEG equivalent for neuronal cells. We use silicon cantilevers to look at a force output, which you can then use to look at your ejection fraction and things like that in terms of the contraction force itself. So that's represented up here. We use microelectrode arrays, which are custom developed, and silicon cantilevers. And we have custom developed hardware that then measures the outputs that I was talking about. So focusing back down here, which gives you the functional outputs, as mentioned, if you look at the silicon cantilevers, we end up getting signals that correspond to a force output, which you can then look at before dosing and after dosing and look at how that force changes. At the same time, if you look at this at the top right, it is essentially the field potentials that you get from the cardiac cells, which if you go to the hospital, you're going to get plugged onto an ECG. It is the equivalent of that, and you get signals similar to a QRS interval and a QT interval, right? So again, an outcome that you can directly correlate to the clinical research that already exists. Now, this would really help with the scaling when it comes to the PK/PD side of things. So moving on to the next slide. We talked a lot about functional measurements and how they are pharmacodynamic outputs and how we like to have those to be endpoint assays that we can compare to the clinic. Now, if you want to have relevant pharmacodynamics and scale them to what happens in vivo, then you need to be able to account for the PK that's happening in your system also. We had some excellent talks before about matching the in vivo PD to the in vitro systems, but at the very least, you need to understand all the different components of the pharmacodynamics in your system if you want to be able to translate those. So as everybody knows in this room, when it comes to the in vivo setting, you have 4 primary factors that are accounting for the concentration over time in the different target sites in your body after a single dose, or essentially any kind of dose, right? So you have absorption, which is the drug passing across your GI, your gastrointestinal tract. You have distribution, which is your heart pumping the blood across your system. You have metabolism by your liver and the GI tract, and finally elimination, whether that be through your stool, kidneys, you sweat it out. But any combination of those, depending on the chemical formulation of the drug, will affect the target site concentration. Now, when it comes to a microphysiological system, what affects the concentration over time, right? Sometimes you have completely different parameters that affect the concentration over time, at least in our experience. When it comes to distribution, you can equate that if you're using pumps, it's directly related to the pressure gradient that you set up or the flow rate you set up. The way we induce flow is using a Rockwell platform to have recirculating flow, and we get an effective flow rate, but we can account for the flow rate, right? So that's distribution. But there are other parameters where in the body, your elimination might be only due to the kidneys and other forms, but in your microphysiological systems, you have all kinds of materials. So a lot of systems still use PDMS and all kinds of other polymers. Pumps use tubing, and the more hydrophobic a molecule, which a lot of drugs are, the more loss that you get. So at the very least, you need to be able to account for them, because if you're not able to account for them, you think you're dosing it with 10 micromolar, but what you might end up with is 3 micromolar, depending on how hydrophobic the drug is. Those are the different kinds of considerations that you want to take when you come to the microphysiological systems. For this project itself, what we wanted to do was look at a platform that recapitulated the barrier tissues primarily. So in this case, we were looking at the GI and the blood-brain barrier to see how it affects the CNS cells. This was primarily developed from the perspective of an Alzheimer's model. So if you look at the middle over here, that shows you a schematic of the housing itself. It consists of 2 different layers, and the GI barriers go over there while the BBB goes over there. So how the system itself was built was it's a polycarbonate membrane that's sandwiched between 2 pieces of PDMS, and they are inserts that you can culture your barrier tissues outside of the system before implementing them into the system. So that also gives you an extra, advantage, because a lot of times you assemble multi-organ systems and you lose one organ, you're screwed, right? So in this case, that's how the system itself was designed. Now moving on to some of the individual organ constructs themselves. So here is the cortical neuron model that was developed, which we use for the CNS side of things. The cortical neurons were developed using human-induced pluripotent stem cells. So what you see here is the protocol that was used for culture, which is available in the publications below. I talked briefly about surface chemistry before. So what we do using surface chemistry is we're able to pattern our microelectrode arrays, where on day 1 you would have only the 2 blobs of cells that are grown there, but over time you see them throw out their axons and connect to each other. So what has been done using this is measure the action potentials, which would be similar to your EEG signals in vivo, and looking at inducing LTP induction, which is long-term potentiation, and seeing how that changes over time. So here it's showing that we don't lose long-term potentiation and that we can induce long-term potentiation. So this is the model that's been included into the 3-organ system. Again, these are the protocols that we use for the BBB and GI currently. But moving on to what those systems look like. Here's the BBB immunocytochemistry as done on the cover slips before they go on into the system. So what you see on the top here are Zo1 and Aclodin, which are the barrier, which are markers for the tight junctions themselves. What you see in the right is GLUT1, which is a marker for the glucose transport, the active transport, which is also located in the barrier junction. What you see below is the marker for the astrocytes. So the model for the BBB itself consists of induced pluripotent cell-derived BMECs and astrocytes on opposite sides of the polycarbonate membrane in this case. When you look at the GI immunocytochemistry, what you see here on the top again is the gap junction proteins or the tight junction proteins here. And on the bottom, you're looking at a marker for the mucin, which is for the mucus. So essentially, the GI itself consists of 3 different cell types: the KCO2 cells, for the representative of the enterocytes, the HT29 for the mucus-secreting cells, the goblet cells, and myofibroblasts on the other side of the membrane. Now, this is the immunocytochemistry of the cells after they come out of the system from the membranes. Again, the same markers, including PGP down there. We talked a lot about functional measurements. So how do we characterize the tight junctions and the functionality of these barrier tissues that I talked about, namely the GI and BBB? What we use is something called transepithelial or endothelial electrical resistance. That's essentially a metric for barrier integrity, and how it works is essentially you have electrodes on 2 sides of a barrier tissue. Usually you can do a 2-point or 4-probe method. We use a 4-probe method, and doing that tells you essentially Ohm's law. You put a current through it, usually an AC signal, you measure the voltage, and it gives you a measure of the electrical resistance across the barrier. So characterizing the systems that we presented before, looking at the very left, you're looking essentially after administration of 3 different drugs: caffeine on the left, bicucaline in the middle, and canic acid on the right side. This we were looking at because caffeine should flow freely between the GI and the BBB. Bicucaline goes through the GI easily, but not the BBB, versus canic acid has trouble getting through the GI, but goes through the BBB freely. The threshold that we had here, looking at transport studies previously, was 1,000 ohm centimeter square for the BBB and 250 for the GI. But essentially, the slide tells you that adding those drugs did not affect the functionality of the cells themselves. Now moving on to some transporter activity characterization. So this slide itself is looking at the GLUT1 that we looked at some immunocytochemistry earlier for. Essentially, the barrier, the cell walls themselves have the membrane, have the transport for the GLUT1, and we use a glucose analog. And what you see here in the first panel is at day 1, 2 after assembly into the microfluidic systems is if you block those GLUT1 transporters, you see a significant decrease of the transport of the molecule through the barrier itself. Similarly at day 7, 8, essentially telling us that the tight junctions are maintained and a lot of the transport is due to the active transporter. Now looking at a second marker for transport, here we're looking at the PGP-mediated transport. On the left here is the immunocytochemistry showing you the existence of those transporters. In the middle is looking at the active data that we collected. What you see here is at the very bottom is just bicucaline alone, which it passed freely through the BBB when dosed on the other side in this case, which you saw that it did. But when you start blocking with higher and higher levels of verapamil, which block the PGP pumps, you see a decrease in the blood-brain side transport down to the blood side. So what we did here was we dosed it into the blood. We dosed it into the brain. That way we can look at pumping back into the blood, which is what the pump itself should do. What we show on the right here is that there was no change in the TO values, essentially showing that the quality of the cell cultures themselves were maintained. Now looking at some of the preliminary PK/PD modeling work that we've done on this, the model that we essentially decided to use was a 3-compartment model, and for the PD part, which is linked to the PD part, we had a GI compartment, a blood compartment, and a brain compartment. There were clearances established between the 2 compartments in the blood compartment, and we established a volume of distribution for each compartment to look at and account for any loss to the materials of the housing themselves. So as you see here, these were essentially the equations defined for the different compartments. They're just basic mass balance equations. Each equation is defined by the amount of drug that you dose it with, the concentration, and the volume. So here's looking at some of the PKA data that we got from using purely acellular housings, right? So this is a characterization of the microfluidic housings themselves. So what we really wanted to see initially was can we see a difference between the different routes of dosing? Since we use the polycarbonate membranes and they have pretty small pores on them, you would see a difference even without any cells. But that needs to be characterized so you can see a difference when you do have cells on them. So what we see here is in the IV dose, which is on the top panel, and the oral dose, which is in the bottom, you see a significant difference in the profiles that we see in the system. So again, this system is to look at, if you want to test different formulations, can we look at is it crossing the barrier or not, as much as not directly recapitulating the whole body PK parameters as we saw earlier before. So again, we looked at this with the 5 different drugs, some of them caffeine, which should readily go through. Now you might ask yourself, why is something like caffeine perfectly mixing throughout the system versus something like nimodipine, you see a lot of loss? There's nothing uptaking the cells, there's nothing uptaking the drug. The drug has nowhere else to go, so where's it going? Right? That's where some of the materials of the housing itself come into play, especially if we use pumps and a lot of tubing, that's even more exacerbated, right? So even without any cells, you see a significant decline in this case. Now, if we don't actually account for this beforehand and we start our dosing experiments, you're going to be way away from where you actually want to be. And at the end of it, you think you have data at a certain concentration, but you actually don't, right? So based on that, this was the first step that we wanted to do, and based on that, these are just looking at some of the goodness of fit parameters, showing that the model was able to reasonably predict the data that we acquired, and looking at the table of the clearances between the blood and GI, between the different dose methods, that is IV versus oral, you see a significant difference with the flow rates being significantly higher. So based on this, we first did it with a rat model, and which we're not going to go into today, right? But looking at the humans and where we are at today, this is looking at caffeine at acellular versus cellular housings, which is what I talked about earlier. It should freely pass through both the membranes, and what you end up seeing is exactly that. They have very similar PK profiles, versus when you look at the IV dose, again, they're pretty similar because you shouldn't see a significant difference. When you come to canic acid now, which we expect the GI to impede, when you look at the oral dosage, that's exactly what we ended up seeing. And then versus when you look at the IV, you don't see a significant difference because it was the IV dose. So this is about the extent we're at here, and right now we're applying the models that I showed you earlier and collecting more data. But overall, in terms of conclusions and future work, we developed a 3-organ microfluidic system with the BBB, GI, and CNS, and we validated the health and the robustness of the cells themselves using different kinds of molecules to look at transport and active and passive transport. We showed that the BBB presented the appropriate tight junction proteins and transport functionality. We developed a 3-organ PKPD, a 3-compartment PKPD model for interpreting the barrier transport and neuronal response. So like I said, currently this model is being investigated with a wide range of drugs, and the model itself will be applied to all of these drugs and to look at the efficacy. Finally, a final set that we're also looking at is including a liver with this model. That way we can also include metabolism. That's a summary of everything that I have today. Thank you.